Thank you for giving us an opportunity to present our company. My name is Jin, an English name, and I'm heading the Finance, Business Development, and Operational Activity and Novelty Nobility. We are Novelty Nobility. I know it's a quite unique name for a new drug development biotech, and it refers to novel science and novel management. It's our daily mantra, speaking to ourselves. And simply, it's like, speaking people around you, I'm gonna quit smoking when you actually try to quit smoking. What we do is we are basically antibody company. So we have a Precise D, which is internal fully human antibody discovery engine, and focus on oncology and ophthalmology indication. And as to the modality, we are using naked antibody as a signal blocker, and also globally hot modality, which is antibody drug conjugate, and also have some proprietary linker technology called Precise L, which is enabling site-specific conjugation and DAL control, and also continuously expanding into the other antibody-based modality, which are multi-specific antibody, immuno-oncology, antibody cytokine fusion proteins. So I'll revisit the Precise D approach at the end of this presentation. So based on this basic setting, our current pipelines are composed as this. So we have a lead item and an N2011 in ophthalmology indication, which are mainly treating retina diseases, and also have some bispecific antibody to supplement some insufficient properties of an N2011. And also, as a second lead item, which is called NN3201 in oncology as an antibody drug conjugate modality. So I'm going to focus on the NN2001 and NN3201 for today's presentation. And I will show some key data and what's key concepts, and then how these programs will be developed in the future. So first one is NN2001. It's novel anti-c-KIT antibody for treating retina disorders. And mainly in this field, anti-VEGF therapeutics are used as a standard of care therapeutics, but we do not focus on the VEGF as a traditional target, but a focus on the c-KIT as a novel target based on our decade of research and mechanism of action. So as you all know, retina diseases are quite vision-threatening eye diseases caused by the growth of abnormal blood vessels in retina, leading to blindness if poorly managed. So the lead indications are wet AMD, age-related macular degeneration, and diabetic complications such as diabetic retinopathy and diabetic macular edema. And as you see, as the disease develops, your sight is going to be gone when you see in the left bottom pages. And then the global market size for therapeutics of retina disease are currently slightly more than $10 billion. And it's growing at more than 4% annually. It's quite large market, and many players are working in this field. Standard of care therapeutics in this field, as I mentioned earlier, are VEGF inhibitors. So there are 3 approved drugs available in this market, which are Eylea, Lucentis, and brolucizumab. Each were developed by Regeneron, Roche, and Novartis. And then in summary, these therapeutics, the standard of care, has a common insufficient issue, which is unmet medical needs in this field, that is insufficient efficacy in the real world. Even though those therapeutics has been improved retina disease treatment dramatically, it has quite a fundamental insufficient efficacy problem. And then some investigators in the world are competing against developing non-VEGF inhibitors as to the combining therapy to the standard of care therapeutics, and also developing long-acting anti-VEGF therapeutics to address the problem of low patient adherence. So sustaining the efficacious period more than 3 months, that enabling to year-long efficacy period sustained. And also, the other group of investigators are developing a non-intravitreal injection type of assets, the oral or eye drop formulation, but these are mostly failed in the global clinical trial because it's harder to make a balance in between the efficacy of delivery and then system toxicity. Having those landscapes set, we're focused on the most SOC, standard of care, and currently investigated clinical trial assets are, requires chronic treatment. Because retina diseases are side effects occurred during the spontaneous recovery of hypoxia. And without resolving the hypoxia itself, if you're trying to suppress the abnormal blood vessels only, anytime your efficacy period is done, the abnormal blood vessels recur, and continuous treatment of anti-VEGF therapeutics is required. That's why many companies are competing against the long-acting VEGF therapeutics. But we bravely argue that we are focusing on disease progression arrest, which is a novel mechanism. And compared with the standard of care therapeutics in this market, our target goal is reaching at the time point beyond that time point, no longer additional treatment is required. So we basically would like to stop the disease progression by making several injections, and then that will ultimately help patients to free from this retina disease chronic treatment. So let me explain this in a little simpler diagram. So drusen accumulation is a root cause for the wet AMD, and glucose toxicity accumulation is a root cause for the diabetic retinopathy. And then even though those are physical root causes are different, anything comes into play that induces hypoxia in eye. And once the hypoxia switches on, HIF-1 alpha, hypoxia inducible factor protein, is stabilizes. And then once the HIF-1 alpha stabilizes, there are two consequential activities occur, which are the glycolysis activation and angiogenic cytokine expressions. And if these two things combine together, as we know, the active angiogenesis occur, and all the SOCs and currently clinical stage assets are treating the abnormal blood vessels only, as I mentioned earlier. But our strategy is basically directly, by inhibition of c-KIT, directly control the abnormal blood vessel and vascular permeability. But at the same time, we have an indirect suppression effect in the much upstream, which is HIF-1 alpha directly. So there are significant benefit in treating the regulating HIF-1 alpha directly, which is more upstream, which are, when you see the left side data in the next page here, by treating 2G4 antibody, which is our NN2001 antibody, the same asset, we can control the HIF-1 alpha and its corresponding pathological VEGF. And then VEGF is well known to be required for the vascular homeostasis, as you know. And when you use the VEGF inhibitor, and then the VEGF inhibitor suppress the VEGF level below to the required level, the additional problems occur, so such as avascular area expansion and long-term blindness. But by controlling the HIF-1 alpha with an antibody that has no binding specificity to the VEGF directly, we can control HIF-1 alpha and pathological VEGF only without impacting on the physiological VEGF, which are required for the vascular homeostasis. And also, this data can be confirmed in vitro study as well on the right side. And then interestingly, the aflibercept anti-VEGF therapy SOC in this field has no impact on controlling effect on the HIF-1 alpha. So that's fundamentally differentiated. So it's a mechanism differentiation. And also in head-to-head comparison, our NN2001 shows quite compelling data set in terms of efficacy and PK parameters. So when you look at the left side data in head-to-head comparison, in the mostly using CMV model, it showed better efficacy. And then PK profile is also superior to Eylea. So with this all scientific data in hand, our ultimate goal is to be fundamentally differentiated from all the SOCs and all the currently clinical stage assets by improving the efficacy and then the durability of the the treatment at the same time. So when it comes to the efficacy side, we can control the most of angiogenic cytokines at the same time by regulating HIF-1 alpha protein, which is transcription factor. We can much improve the efficacy. And also to the durability. As I showed the earlier page, it's a property-based durability. It's much better than the EYLEA, but there is also a mechanism side durability. So by controlling the glycolysis and angiogenic cytokine at the same time, we can remove all the energy source and the material for the active angiogenesis. So in the clinical trial, we'd like to confirm if this asset can extend the efficacy and durability at the same time. So, as to the timeline, we are conducting a GLP toxicology study right now and targeting to file an IND submission to FDA early next year. So the first in-human study trial will be conducted next year in the United States, and then will be expanded into the other retina diseases in the 23. So please take a look at how this program can be progressed in the future. The next item is NN3201, which is a novel anti-c-KIT antibody drug conjugate program for treating solid cancers. So c-KIT in oncology is a quite meaningful target, but not massively investigated previously. We especially interested in GIST, gastrointestinal stromal tumor, and SCLC, small cell lung cancer. And then in GIST, c-KIT mutations are well known to be a driver of that indication. And then mostly 85% of patients are somehow associated with the c-KIT mutation, and additional 5% of the c-KIT wild type. But the standard of care for the GIST is an imatinib, which is quite a nice drug, but its efficacy is quite limited, and then does not really kill the tumor before getting to the resistant mechanism. And when it comes to the SCLC, small cell lung cancer is a subtype of lung cancer which accounts for 15% of entire lung cancer patients. It's quite aggressive cancer, and the current standard of care therapeutics is a PD-L1 therapeutics plus chemotherapy, which brings about 8 months overall survival. Interestingly, PFS does not have any meaning in this indication because it's quite aggressive, and then people are going to be dead before reaching out to the PFS. So usually the OS is much more important, and PFS and OS has no correlation in this indication. And c-KIT, as you can see in the table data, is one of a very few target that has a positive correlation with the overall survival in the SCLC patient. And then more than half of the patient are c-KIT overexpressed. So we are targeting these patients with the ADC form. So due to the time limitation, I did not bring the specific data of how this ADC is composed of. It bears a proprietary linker technology which enables site-specific conjugation and DAR control. Its ex vivo stability is much better improved than the commercial linker, and also use the commercial payload in consideration of this, the target characteristics and the indication specifics. So let me show some key efficacy data here. In the GIST xenograft model, our ADC reached the complete remission at the 1.5 mpk dosage level, and it's above. So it's quite amazing data set, and then without any concern in toxicity, at least in the xenograft in vivo study. And more attractively speaking, the SCLC, which is much more aggressive, and then no targeted therapy available for the SCLC, but with this ADC monotherapy from the 3 mpk level and its above level, we also reached the complete remission in the xenograft model study here. And then that data also has no toxicity concern. So we think this data is quite meaningful, and then setting up the very baseline of our therapeutic regimen strategy. And also preliminary toxicity has been tested massively. So up to 60 mpk level in vivo, the drug was quite well tolerated. And so the therapeutic index, which is the most important parameters in the recent ADC development, is more than 20 times, and then it's quite well tolerable and then well efficacious at the very small dosage level. So with this research data in hand, we are targeting to ultimately change the therapeutic regimen for the GIST and SCLC. And first, the GIST setting, as you see here, Gleevec is the first line of therapy, and Sutent, and regorafenib, avatinib, repretinib are being launched in the market. But in combination with the Gleevec, we target to increase the median progression-free survival by 2 times, double the times. So from the 1.5 year previously with the imatinib alone to at least more than 36 months with the combination therapy with our ADC. And in SCLC, therapeutic regimen strategy is currently being developed, and much more research work is being conducted internally. So as to timeline, we just made a candidate selection decision in partnership with our CDMO partner globally. And thanks to their help in our collaboration, we will go into the scale of manufacturing in the second half, and also IND enabling study by the end of this year to prepare the IND submission by the end of next year. So these 2 assets are on the market to search for the right global development partner. So if you're interested in this asset for any type of partnership, we're quite open. We're flexible to make any type of collaboration structure. So I have about 10 minutes remaining, and then I'm going to speak about a little different story. So all based on this fundamentally differentiated and outperforming assets, the core is our fully human antibody discovery engine, which is called PRECISE-D, and then D refers to design and discovery. And I'm gonna spend another 10 minutes explaining our fully human antibody discovery approach, and then how that can be benefit to all of you. So even though we are antibody-focused company and we have some enough internal capability of the fully human novel antibody discovery, we start from a question not from the technology, but from our more philosophy-oriented question, which was, what is good antibody? So with the 2 decades of hands-on experience and then all the ground job, my CEO, Sang-Kyu Park, who is an incumbent professor at Aji University, has a quite decent conclusion, which is a good antibody is a fit-for-purpose antibody. So recently, as an ADC form, as a CAR-T form, as a TCR-T, and then whatever the other things like a pseudo-antibody part, the antibody has a multiple function and multiple applications. So initially, it was used as a signal blockers only, but nowadays, its usages can be much expanded. So it is quite important to understand what's the purpose and then what's the potential applications before going into the discovery action. So that's why we think the good antibody is a fit-for-purpose antibody. So technically speaking, PRECISE-D platform is a discovery of fully human antibody leads in 4 months by using humanized mice platform, HuMice platform, so-called. And then we currently use Hubble Biomed H2L2 mice. But interestingly, we do not own any technology platform for the antibody discovery because we want to be agnostic and we have sufficient flexibility and move on to the next best target platform technology if the new technology is introduced in the market. And then at this moment, we are often asked question about, okay, what's the difference between the library screening approach and HuMice used approach? So HuMice used approach is immunization-based approach. And our short answer is, there is nothing like one is better than the other. and each technology has its pros and cons. But I'd like to focus more about why we selected the immunization-based approach. So conceptually speaking, phage library screening is more like, using a fishing net for fishing. It's like using a shotgun for aiming. So it's basically collecting some fraction of antibody from a pool and then assembling those into a full antibody form. Rather, immunization-based approach using a humice or maybe in vivo model, or like using a harpoon for fishing. It's like a sniper approach. It's aiming to the head and then making a headshot. But the problem here is it's much more harder to control because it bears a much higher level of variability. So when you think about the normal distribution curve of its output, immunization-based approach can bring much more broader range of outputs. So the key is how you can actually control the higher level of diversity in order to get the extraordinarily, it's outlier-like, high quality antibody, which locates at the extreme bottom of that normal distribution curve. So that's where we have some accumulated know-how and knowledge for the past 2 decades. So as to the process, it starts from the very common starting point, which is understanding unmet medical needs. And also, we select a drug target very seriously based on their unmet medical needs. But there are 1 or 2 more steps here, which are, we think about the modality strategy first. So because based on what modality you'd like to pursue, there are required properties can be defined differently. So having all this modality strategy, medical unmet needs, and drug target selection all together, we really focus on design of the antibody. So immunization-based approach, in short, is basically about design and realization of the designed property in the actual discovery phase. So more specifically, we focus on the mechanism of action, whether it can be a signal blocker or the delivery tool, and what epitope binding site is proper for that specific strategy. So certain membrane protein target has a different function per epitope binding site. And also we have to consider whether we need to immune system involved or not. If signal blocker, we should bind to ligand side or the receptor side, and whether it really requires a competitive binding. And the most target as a signal blocker requires a competitive binding. And if delivery tools, ADC versus CAR-T, I just bring about the example. But based on our experience, ADC efficacy is possibly correlated with its binding affinity, but CAR-T requires a relatively lower level of binding affinity because it needs to deliver the T cell to the target and detach from the target and then deliver another T cell to the target. So the required property for the antibody when it comes to ADC purpose or CAR-T purpose can be different, and then we need to specify these things before going to the discovery. And then when it comes to antibody design part, it's important to consider what type of antigen will be optimized. So whether we need to use a protein or a peptide or maybe the other things, target structure-based recognition with the help of in silico approach, and antibody type, IgG or IgM or maybe probably IgG1 from 4. It all has different functionality. Effector function is much more important, area of research in these days because some target indications and really requires improvement of the effector function, ADCC or CDC, to improve the anti-tumor effect, while the other indications, too much improved effector function can bring about some inflammation-related adverse events. And then we prefer to use a native antibody without an engineering, but have also internal capability to use an engineering if required. But generally speaking, do not prefer to use engineering because antibodies are kind of, dynamic biologics. And then when you're trying to change some part, the whole balance will be broken. So for that reason. We usually prefer to set up the screening criteria before going into the discovery action. And then once all these things are set up, the actual discovery will take only 4 months to get the antibody leads, the multiple, and then currently use Harvard Biomed, as I mentioned. So this one will be service available from 2023. The order to discover service we're preparing internally. And this year and the next year, we have much antibody targets to discover. And then once we discover all the required target internally, we will provide some order to discovery service to the other biotechs in Korea first. So thank you for listening to my presentation. And any questions from the audience?
Website.
Yeah, website. Okay, thank you.